Hello and welcome to the 1516 Biology Unit 8 Genetics and Heredity Curriculum Overview. As we take a glimpse of our instructional focus document for Unit 8 Genetics and Heredity, TRS suggested a duration of 15 days. However, on our GPI's DAG, we have been allotted 16 days for the unit. Our unit will begin when we come back from holiday break on January 4th through January 12th, closing out the second quarter of our year, which is the end of the first semester. Now, the second semester will start with quarter 3, and we'll pick up this unit on January 19th through the 29th. As you see below, our processing skills are in black. They are noted in black, and our supporting standards are in green, and our readiness standards are in red. For this unit, this unit introduces our readiness standard B6F and the supporting standards B5C, B6D, G, and H. This is the last time B5C, B6, B, D, F, and H are addressed before the STAR biology assessment as far as the entire unit goes. Of course, you want to make sure that you're spiraling in as you continue throughout the school year. Now let's look at our prior to this unit. I'm going to go in further detail about the prior to this unit and during this unit later on in the presentation. However, we want to make sure that we are incorporating our prior to the unit as the development of our engaged activity. During this unit, we utilize this for our explore activities in the explain portion of our 5E model, as well as our elaborate and evaluate. And after this unit, the students will be able to utilize these concepts in genetics through the reinforcement and building of units on evolution and studying the kingdoms of living organisms. So after we've viewed our unit overview and gone through our instructional focus document, we want to look at our district level data to see how we have performed, as well as look at your individual campus data to see how well you all performed on the Unit 8 in the prior years. And so as for the district level in 13-14 on the Q3 summative exam, we see that we've been pretty stagnant on our SEs for B6D, supporting standard, B6F, readiness, B6G, and B6H, which are both supporting standards. In 14-15, we haven't really demonstrated much growth in these SEs, so we know that these are SEs that the students struggle with in understanding the concepts of genetics and heredity and all that it encompasses. So we make sure that we are spiraling in as we continue on throughout the school year and the additional lessons or units that are coming up. Because these are critical SEs that you will more than likely see assessed on the biology STAR. I have notated asterisks by these 4 SEs simply because these are the newly introduced SEs that we've never seen in the school year, and these other SEs have been introduced in previous units. Looking at our STAR data, and you can look at each of these student expectations for each of the STAR years and notice that 2015 was the year where we excelled in the bulk of our SEs, with those SEs really being the supporting standards, so that we can see a lot of emphasis were placed on the supporting standards based off of the percentage over 70% on those SEs. However, our readiness standards B6A is at 54%, and our readiness standard B6F for the unit, which is newly introduced in this unit, is at 47%. So we want to definitely make sure that we are putting just as much emphasis on these readiness standards as we are on the supporting standards. So we want to make sure we're demonstrating growth in all of our SEs, but definitely especially in our readiness standards. As we look at how the state has tested these SEs, we will look specifically at the SEs that are newly introduced in this unit And for the supporting standards, there will be one question for each. And for the readiness standards, I will provide 3 questions, one from each year, 2015, 2014, and 2013. So looking at our first SE, B6D recognized that gene expression is a regulated process. And I have deconstructed the actual SE over here to the left. I circled my verb, which is recognize. I have put an underline under the content that we're focusing on, which is the gene expression. And in what context are we focusing on gene expression? As it is a regulated process. And so what does the term or verb recognize mean? Recognize means to identify something previously seen, known, etc. To identify from knowledge of experience or characteristics. And that comes from the Kilgo 2015. And as we look at the question is simply asking, how has the pigment changed in the body of the rabbit since the temperature has changed? And so the students will have to understand that gene expression is taking place and it's being regulated by temperature. At one temperature, the bodies are white. At another temperature, the bodies will turn black. And so we want to be sure that we understand how the state is assessing this SE so that we can effectively develop our explore activities throughout our lesson cycle. Now looking at our readiness standard B6F, predict possible outcomes of various genetic combinations such as monohybrid crosses, dihybrid crosses, and non-Mendelian inheritance. And now again, I've deconstructed the SE. Predict, this is our verb. What does predict mean? To state, tell about, or make known in advance, especially on the basis of special knowledge. So we're going to predict possible outcomes of various genetic combinations, and this is our content, possible outcomes of various genetic combinations in our context, such as monohybrid crosses, dihybrid crosses, and non-Mendelian inheritance. And I want to make sure that we all understand what such as means in the SEs. And such as is stating it's an example. If it says including, that means specifically these things. But in context of such as, these are different ways you may see it. And so here in the question, this is a dihybrid question. It's focusing on 2 traits, dimples and earlobes. And so the students will have to do what exactly it says to predict trends. That's why this is a dual quoted question for 2G. And 2G is analyze, evaluate, make inferences, and predict trends from data. And we want to be sure that we're having the students to practice and experience predicting and understanding how to analyze and determine what those trends are from questions. Now another question from 2014 over B6F. Again, it is dual coded, and this time we're focusing on monohybrid crosses. So we have to, again, be able to do it in multiple ways, looking at monohybrid, dihybrid, as well as non-Mendelian inheritance. And this question is from 2014. And for B6F in 2013, again, it's dual coded, same processing skill. Analyzing and predicting those trends, and now they have to determine which of the following genotypic ratios can be expected from the genotypes that are predicted, the alleles of black coat color, which is dominant, over the allele for brown, which is recessive. Now looking at our next SE, B6G, recognize the significance of meiosis to sexual reproduction. Our verb recognize, our content, the significance of meiosis, and what context is to the sexual reproduction, and I have it here notated for you. We do know what recognize means from the previous SEs. This is a supporting standard. It has not been dual coded for the 2015 STAR assessment. And here the students will have to read this question, look at the number of chromosomes in a nuclei, and understand that meiosis produces sex cells or gametes with half the number, and so the answer here would be 23 chromosomes. And last but not least, our last SE or focus, B6H. Describe how techniques such as DNA fingerprinting, genetic modification, and chromosomal analysis are used to study the genomes of organisms. And one thing for sure that we know, again, our verb is describe. Describe means to give an account of in words, to tell in words what something or someone is like. And we're describing techniques such as DNA fingerprinting, genetic modifications, and chromosomal analysis. And again, these are just examples of techniques. There are several others, and you will see those noted in your specificities. Are used to study the genomes of organisms. And the one thing, in order to study a genome, the students will have to know DNA has to be present. And so as they look at this question, they have to understand how the genomes of people with a condition or without a condition can be studied. And if they retrieve samples from blood samples or cheek swabs from people, what has to be in common and what must they both contain? And the answer here is a complete set of DNA. And this is a dual coded question as a supporting standard, 3D, and 3D means to evaluate the impact of scientific research on society and the environment. Now, for the sake of time, I will not go through and read all these previous PAs for the IFD. However, I do want to make note that we are using the 1415 GPIC modified PA for Unit 8. There's only one PA that has been comprised of all of the previous PAs, PA 1, 2, and 3 into one. And so please make sure that you are utilizing the modified PA for this assessment. Now, one thing I do want to note is that the following techniques, you can address several other techniques and look at other questions and see what techniques have been assessed in the past, and just make sure you're incorporating all different types of techniques to be studied and explored in your activities. And we have a quick snapshot of our modified PA with some additional specificities for each of the SEs. I don't have all of the SEs on here, but I just wanted to show you a few that we've touched on. B6D, this one here is B5C, and this one is B6H. And again, use these specificities as you are also following your IFD overviews during the units and incorporating all of these different aspects into your lessons. Now, what I've done here is I have developed a compilation of all of the essential questions that were provided for each of the PAs. And so we have all of the essential questions here from all three of the PAs into one for that one PA. And so these questions, please incorporate them throughout your lessons, put them in your explore activities, make them a part of your explain specifically, and in your elaborate and your evaluate as well. Because this will help you to see if the students understand the big picture and can they explain what they have learned from all of the activities that they've done. So these will definitely help you. You don't have to think of them. They're already provided for you. And a few of the misconceptions we definitely want to make sure we are touching on and addressing in our instruction, especially about Punnett squares. Students may think that they give exact ratios, and they don't. They're only providing a probability of a particular outcome. Students think that dominant traits are more common in populations than recessive, and that's not true. You can have an equal amount of both, or you may have recessive over dominant, but there are variations. Students may think that all or most traits follow the Mendelian laws, and that's a misconception as well, because all factors are not really explained by the Mendelian laws. There are some affected by other means, so we want to make sure we're addressing that. And students may think cell differentiation is a result of loss of DNA or genetic material, and it's not. It's due to gene expressions which causes cell differentiation. And then we have our vocabulary that the students will be exposed to and experiencing throughout our 5E lesson cycle. In addition, a slew of related vocabulary that you can find as well in your IFD. So as I previously mentioned earlier in the presentation, we were going to go more in depth about the prior to this unit and how to develop your engaged activities. And so here, as we can see, I've read through it. I can see that the students have been exposed to genetic instructions or genetic information and understanding how instructions go from one generation to the next and how they inherit traits, which is governed by genes in the DNA. And so we want to definitely be sure to incorporate the knowledge that the student should have from previous grade levels as well as what they have done in previous units in biologies, because we want to make them understand and see the big picture and hopefully get them to grasp the concept that all of these aspects of biology are connected and they can then see the big picture at the end. And so definitely spiral in biochemistry, definitely spiral in protein synthesis, and use those activities or use this information to develop your activities that will then make the lesson flow even easier so they can understand how protein synthesis now relates to the transfer or the development of traits. Then during this unit, one thing I wanted to express is the way they have written during this unit is really how you should make sure your explore activities are flowing, because how they write it makes the connections between what they should understand before they understand this aspect or before they understand the relationship between this next aspect. So as you read through it on your own, you'll understand how they're going to go from their engage of what they should know to what they are learning during this unit. And when they finally end on their last activity, this is what they should be able to tie in back to the scientist Gregor Mendel and what he explored in understanding the field of genetics and recognizing how genetics has advanced from that time to now. And lastly, to close out, some important reminders. You can see again, STAR accommodations are available on Board Builder, which you will find on Discovery Ed. We have SST. Please contact me directly to receive the join code because it does change often. Exemplar lessons, activities, rubrics, you can locate them on the Secondary Science Shifter Resource Dropbox. There will not be a Q3 3-week or Q3 6-week district assessment. We will have the Unit 8 assessment for being assessed on Q3 summative, which will be held March 2nd through the 4th, 2016. If you have any questions or you need anything, please do not hesitate to contact me. Again, you know how to reach me. I'm Erica Ingram, and I hope that this curriculum overview has helped you. Have a great day.